The cardiac cycle is a series of coordinated events involving pressure changes, volume changes, and valve movements that occur each time the heart beats. Systole refers to the contraction of the chambers, and the diastole refers to the relaxation of the heart. Unless specified, systole and diastole refer to the ventricles. A pressure-volume loop is a graphical representation of the cardiac cycle that moves time from the axis and focuses solely on ventricular pressures and volume. Looking at the pressure-volume loop, we can trace the cycle. There's the diastolic filling. This phase begins at a point VF, when the mitral valve opens because the ventricular pressure has dropped below atrial pressure. Blood rapidly fills the ventricle passively during approximately the first one-third of diastole, with the aortic valve closed. As the ventricle fills, the volume change slows. In the example shown, volume increases from around 50 milliliters to 120, with only 10 millimeters of mercury increase in pressure. Point M represents the closure of the mitral valve. Then we have isovolumetric contraction. As systole begins, ventricular pressure increases rapidly. When ventricular pressure exceeds arterial pressure, the atrioventricular valve closes, which is responsible for the first heart sound. Since the semilunar valve is still closed because ventricular pressure is less than the aortic pressure, pressure increases rapidly, but there is no change in volume. This phase is brief and ends when the ventricular pressure exceeds aortic pressure. When we talk about ejection, once ventricular pressure is higher than aortic pressure, the aortic semilunar valve opens and blood is ejected from the ventricle into the aorta. Pressure increases during the first part of this phase, reaching a maximum of approximately 120, and ejection is rapid. As ejection slows, pressure starts to decrease in both the ventricle and the aorta. In isovolumic relaxation, as the beginning of diastole, ventricle pressure drops rapidly below aortic pressure, causing the closing of the aortic valve at point AC. The closing of the aortic valve is responsible for the second heart sound. Since ventricular pressure is still greater than arterial pressure, the arterioventricular valve remains closed, resulting in a brief period where the volume doesn't change but pressure drops. Once ventricular pressure drops below arterial pressure, the anterioventricular valves open, ventricle filling begins, and the cycle will restart. When we talk about inotropy, inotropy is also known as contractility, refers to the force of contraction generated by the heart muscle. Since all cardiac fibers are activated all the time, the heart modifies strength by changing individual fiber contraction strength through the regulation of calcium. In the pressure-volume loop, entropy is represented by the end-systolic pressure-volume relationship line. This line represents the pressure-volume relationship at maximal contraction of the cardiac fibers. The slope of this line gives an idea of the contractility or calcium regulation during cardiac muscle contraction. Increased inotropy means a steeper slope of the ESPVR line. When we talk about afterload is defined as the force against which the heart must pump to expel blood in the vasculature. It represents the resistance to blood leaving the ventricle during the ejection phase. Afterload is the sum of elastic and kinetic forces that oppose ventricular injection. Essentially, it is the resistance that the heart muscle must overcome to move blood into the stretchy elastic arterial system. Increased arterial elasticity is a common contributor to changes in afterload. Arterial elastance is distinct from ventricular elastance, and it refers to the stiffness of the arterial tree. There's lots of things that can affect our pressure-volume loop, and that's just one of those things. If we increase afterload while inotropy stays the same, it will lead to an increase in pressure during the ejection, but with a decreased stroke volume and an increased blood remaining in the heart. And when we talk about preload is best understood as the amount of stretch in the cardiosarcomeres before they contract. This passive stretching occurs during diastole as the ventricle fills with blood. The volume of blood in the ventricle on the x-axis of pressure-volume loop is when isovolumic contraction begins and is a good indicator of preload. Assuming inotropy and afterload remain constant, an increase in the amount of blood returning to the heart leads to a greater end diastolic volume, thus increasing preload. This increase in preload results in a substantial increase in stroke volume and ejection fraction. Preload is impacted by mechanisms that pump blood back towards the heart or things that have to do with venous return. As veins contain one-way valves and have very little pressure, the skeletal muscle pump, where contracting muscles squeeze the veins, and the respiratory pump, while breathing movements create the pressure gradients to force the blood back to the heart. We talk about mean arterial pressure, or MAP, and how it's regulated. Mean arterial pressure is regulated and it needs to be maintained within specific ranges for normal cellular functions. This is something that is tightly controlled by a homeostatic mechanism to ensure adequate blood flow to vital organs like the brain or the heart. Therefore, MAP is regulated by controlling heart rate, stroke volume, peripheral resistance. The body controls the variables in these equations to keep blood pressure within the homeostatic range. When we talk about baroreceptor reflex, the baroreceptor reflex is a key mechanism for short-term regulation of blood pressure. It is a reflex arc consisting of 5 components: receptors, afferent pathways, integrating center, efferent pathways, and effector organs. If there's a sudden drop in blood pressure, the baroreceptors are stretched less, resulting in a lower frequency of action potentials sent to the cardiovascular centers in the medulla. The medulla interprets this as a drop in blood pressure. In response, the cardiovascular centers decrease parasympathetic activity and increase sympathetic activity. The combined effects increase cardiac output and increase peripheral resistance to help restore blood flow to the brain and increase blood pressure back towards normal. If there was a sudden increase in blood pressure, the opposite events would occur. The baroreceptors would be stretched more, sending a higher frequency of action potentials to the medulla. The medulla would decrease sympathetic activity and increase parasympathetic activity. This would result in decreased heart rate and strength of contraction and vasodilation of systemic arterioles, thereby lowering blood pressure back towards normal. When we talk about capillary exchange is the process by which water and dissolved substances move between the blood in the capillaries and the surrounding interstitial fluid. The interstitial fluid is the clear liquid that bathes the body's tissues. This exchange allows cells to receive necessary substances like oxygen, nutrients, regulatory chemicals, and electrolytes, and to get rid of waste products like carbon dioxide and acid. The movement of fluid across the capillary wall is driven by 2 main types of pressure: net hydrostatic pressure, which tends to push fluid out of the capillary, and net osmotic pressure, which tends to pull fluid into the capillary. The difference between these two is the net filtration pressure. The 4 main pressures involved are blood pressure, or hydrostatic pressure of the capillary, blood colloid osmotic pressure, or oncotic pressure of the capillary, interstitial fluid pressure, hydrostatic pressure of the interstitial fluid, or interstitial colloid osmotic pressure, or onotic pressure of the interstitial fluid. So how do these forces differ at arterial ends versus venous ends? The forces change in magnitude along the length of the capillary. Blood pressure decreases significantly as blood moves through the capillaries due to friction. As the arterial end of the capillary bed, blood pressure is around 30 millimeters of mercury. At the venous end, blood pressure drops to around 10. Blood colloid osmotic pressure, or BCOP, remains relatively constant along the capillary because plasma proteins like albumin do not cross the capillary wall. Interstitial colloid osmotic pressure, or ICOP, is generally low and remains relatively constant from the arterial to the venous end. And our last one, our interstitial fluid pressure, or IFP, is also generally low and constant. Normally there's a slightly more fluid filtered out as the arterial end than reabsorbed at the venous end. The excess fluid that remains in the interstitial space is picked up by the lymphatic vesicles and returned to the circulation, maintaining a balance. When we talk about an edema is an abnormal accumulation of fluid in the interstitial space. It occurs when the careful balance of the 4 capillary exchange forces is lost. An example of this is hydrostatic edema, which happens due to an increase in the hydrostatic pressure inside the capillary. While precapillary sphincters normally regulate flow into the capillaries and maintain relatively normal hydrostatic pressure even with high blood pressure, prolonged systolic pressure above 160 can cause more blood to pass the sphincter than normal, raising capillary hydrostatic pressure and leading to fluid accumulation in the interstitium.